Hello friends, welcome back to my channel Xgene and proteinX. In this tutorial video I am going to show you the Dam and Dcm methylase, and you know very well the importance of this methylase enzyme. They have the main function of this enzyme to add the methyl group in the bacterial sequence, and this kind of enzymes are very much important to protect their own DNA from the cleavage of the restriction endonuclease. So basically the restriction endonuclease, their jobs are to recognize the sequence, and they just cleave the sequence, whatever the sequence it is, the any kind of sequence, foreign DNA or their own DNA, they can cleave their DNA if there is any desired sequence is there. So here the methylase enzyme, they actually add the methyl group and they distinguish between the foreign DNA and their own DNA. So the restriction enzyme, they recognize the sequence, but because of this methylation they won't cleavage their own sequence. So and here there are different kind of the methylase enzyme are available. Mostly three important enzymes are available, and two are very important. That's our DCM and DM methylase. And for the DM methylase, every methylase they have their own, and there are different kind of the characteristics. And as the name suggests, you see here the DA. This A is for, it represent the adenine sequence. So here you see, if you look at this structure, so this is the adenine and this is the thymine. So there is a linkage in between adenine and thymine, and this DAM methylase, their characteristics is they add the methyl group in the N6 position of the adenine sequence, and their specific sequence they recognize, that is the GATC, this sequence. So they recognize this sequence and they add the methyl group in the adenine group in the N6 position. So here is the, you see here, this is the purine, and here in the adenine there is the 1, 2, 3, 4, 5, 6, and here it attaches with the N. So here is the N6 position, and here the DAM methylase, it attaches the CH3 group, and the CH3 group, it transferred from the S-adenosyl methionine, S-adenosyl methionine. So this is basically in short it known as SAM, S-adenosyl methionine. So the methyl group, it transferred from the S-adenosyl methionine. So this is about the DM methylase, and also the DCM methylase. Their jobs are same, they're similar kind of jobs like adding the methyl group, but their characteristics are a little bit different. So as the name suggests, the C, you see the C represent the cytosine, and here DCM methylase, they only add methyl group in the cytosine residues and in the C5 position of the cytosine residues. So here you see the cytosine and guanine, there in between the linkage, and also here you see this is the pyrimidine structure, and here the numbering is from here 1, 2, 3, 4, 5. So here is the C5, and here they attach CH3 group, and they also have the specific sequence they recognize. That sequence is C C A G G, this sequence, and also they have another sequence C T G G. So this both sequence they can recognize, and they add in the cytosine group of the C5 position in the methyl group. So this is about this Dam and Dcm methylase, and also these are very much important in the bacteria. They synthesize this kind of methylase enzyme to protect their own DNA from the cleavage of the restriction enzymes. So this is about this kind of all these methylase enzyme. So if you have any queries regarding this kind of methylase enzyme, you can write in the comment section, and kindly hit the like button, share it, and subscribe my channel. Thanks guys.